Okay, hello everyone. Yes, so I'm Maxwell. I'm a PhD student in the lab of Ulrich in the University of Cambridge, or a PhD student for a couple more months anyway. I'm in my final year. And I have the honor of giving you the last talk of today. The rest of the day may have been long. I hope this talk doesn't feel too long. And I'll be talking to you about, I'm going to talk to you about our first structure-based DNA data storage device. We've heard a lot about sequences. I'm going to tell you about the other side of the coin today. So I'll start with something that you've seen a million times already, which is this pipeline for the DNA sequence data storage approach. So I won't spend too much time on it. I won't labor the point. I will just draw attention to the shout out here to Robert Grass's synthetic fossils. I'm a big fan of those. And there's been lots of excellent progress, as we've seen, but we've also heard a lot about the bottlenecks. So the bottlenecks are primarily in synthesis, which still is done primarily by phosphoramidite chemistry, and in the sequencing. And we would like to find an approach that doesn't rely on the conventional technology for both these things. And the approach which we use in my lab is DNA origami. So DNA origami leverages, instead of leveraging the sequence primarily of DNA to store information, it uses its structure and uses specifically its programmable binding and folding. So in DNA origami, you might be familiar, but you start with a scaffold DNA. So in our case, we use a viral genome, the genome of M13, and you add some staples, which are oligos that you've usually had synthesized and ordered, and they are complementary to different parts of this scaffold. So a single staple has complementarity to different parts of this scaffold DNA. And so what you do is you heat the whole thing up above the melting temperature of all the secondary structure, and then you cool it down again. And these staples determine the shape that it folds into as you cool back down to room temperature. So it finds this thermodynamic minimum that you've designed. And this has been used to great effect to make many beautiful and functional structures. So we have two-dimensional and three-dimensional DNA origami structures here. I've actually been very naughty and I've omitted the references for these. These figures are from Day 21, June 19, and Yao 20. And if you'd like to find those papers, then come to me afterwards. I don't want to snub anyone. But if you think about it, each of these structures in some way encodes information. Or at least if I received this in the post, I would assume someone was trying to tell me something. And the second thing I'd like to draw your attention to is that each of these structures and each of these structures, they all use the same scaffold DNA. So the scaffold is the same, and the difference is in what kind of staples you add to them. So in effect, the information writing here is done by the choice of which staples you add to this scaffold. So structural data storage has a few key benefits which I draw your attention to. So it was used here in this paper using a fluorescence readout. So we've seen, with sequence-based data storage, you're always required to do some synthesis at the end of the day. Some sequencing. But with structural data storage, it has multimodal readout. So this is just one example. You can use fluorescence. You can also use AFM, as we saw in the other slide. It's in principle rewritable. So we can use mechanisms such as toe-hole-mediated strand displacement to replace things that are already bound to our structure and rewrite data that's already been written, which is pretty tricky for DNA sequences. And you have access to higher order encoding. So binary has 2 states, DNA has 4 states per symbol. In principle, for instance, with nanopore sensing, which I'm going to talk about again in a second, you can have as many different states for your symbol as you can distinguish current levels. So you can go up to as high an order as you like, really. So I'll talk a little bit more about nanopore sensing, because what I say after this will hinge on it. It's my preferred method for readout, and in Ulrich Kaiser's lab, we use it a lot. These aren't the same nanopores as you've probably heard of in the context of sequencing. Similar principle, but those are biological nanopores, whereas in our lab, we use solid-state nanopores. So often these are made from silicon nitride membranes with holes drilled in them, or in our case, we use glass nanopipettes. But the basic principle is you have a hole in some kind of electrolyte, and you have a voltage fixed between the 2 sides of this hole. And because of the voltage that's applied between these 2 sides, you have a certain baseline ionic current. So it has a certain resistance to ionic current, and it allows a certain amount to pass. But the fun thing is that if you have some kind of charged analyte, which is pulled through the pore by just electrophoresis, then that changes the resistance characteristics of this pore, and it produces a current blockade. or it produces a change in your current which you can measure and which feeds back on the size of the obstruction. So a larger one might give a bigger obstruction and a bigger nanopore signal. And you can use this to make DNA origami structures like this one here, which is, well, it's a bit rich to call it DNA origami, really. It's a linearized M13 genome, and then it's been duplexed selectively so that it's just a double-stranded DNA between here and here. And then at the ends here, we have some DNA structures that we've added. So these are DNA dumbbells. And these dumbbells in that region, they effectively double the diameter of the DNA, and they cause a bigger obstruction. And so you end up with a nice signal like this, where you have your baseline level, and then the DNA level of the main duplex. You have these subsidiary spikes, and then another one at the end there. And we can think of this region in here as being like a writing frame. So you have these other structures, which you can use as a reference, 2 on one end, 1 on the other, tells you which way it's passed through the pore, because in principle it can travel through in either way. But in the context of DNA structural data storage, let's talk about the pipeline, kind of draw the comparison to the one we showed earlier. So I'll spend a little bit more time on this one. Your encoding will involve turning your data into some kind of structural signal, so a sequence of spikes, for instance, in nanopore sensing. You have to find a way to do this mixing. This is still a problem in structural data storage. Just because you've gotten rid of phosphoramidite chemistry doesn't mean that your problems are over. You still have to find a way to do a lot of mixing, and with ideally not too much material. As Gemma drew attention to, you don't want to be using too much material if you can avoid it. So typically this will be done with microfluidics. Annealing, so as I said, you have to heat the thing up and then cool it back down so it can find its thermodynamic minimum. A useful step is you'd like to be able to pull these things together, so to put all of your assembled structures together so that you can read them out or, in fact, purify them in parallel. You read them, for example, with a nanopore, and then you decode the signals that you find back into your original data. And I've spent my PhD working on different parts of this process. Primarily, I've worked on the encoding side of things, so I've looked at different types of marker that can be used in this multimodal readout scheme to indicate bits. And I've also worked on the mixing and assembly side of things. So I've worked quite a lot in continuous flow droplet microfluidics. I've designed some electron microfluidic devices. And it was in the context of developing and testing this that I discovered an issue with DNA structural data storage, which would come up in the practical applications of it, which is what I'll talk to you for the rest of the presentation about. So there's 2 considerations that we would like our DNA data storage hard drive to satisfy. So first of all, in the mixing step is you should be able to write n bits with at most n oligo additions. This is theoretically the most you can do. If you have to use 2n oligo additions to write n bits, then whatever scheme you're using to mix your oligos together is going to get more and more complicated, and it's going to increase the footprint of the device, increase the number of implementation issues. So ideally, we would achieve this benchmark of n bits for n oligos. And assembled DNA hard drives, they shouldn't be overwritable at room temperature. So after you've mixed everything together and annealed it, we should be able to freely pool those without the leftover writing oligos that are left over from all of these steps here, overwriting everything so that you will end up with ones. And I know this will happen because there are versions of this of a DNA hard drive where it isn't. It is overwritable at room temperature, and I'll show you one of those now. So the most naive overhang-based DNA hard drive, and this is one we've used in our lab. Actually, it was there's a paper for this from Kai Chen, who previously was in our group. He's not naive, but the scheme could be called naive. You start with a backbone, your M13 backbone, a set of overhangs, dumbbells, and complementary oligos, which is going to be the same for all of your structures. And then you selectively add some biatynylated labels. So in this case, we've added labels that are complementary to the second overhang here, B, and the fourth, D. You anneal that all together, and you make this nice DNA hard drive with these biatynylated labels attached at B and D locations. You can add monovalent streptavidin, or any streptavidin if you can use enough of it, and then measure the thing in a nanopore. And you get a set of signals that kind of look like this. And I've used this shorthand structure here, which I will refer back to again. These blocks are just the references, and then the circles are either filled with black for a 1 or left white for a 0. But you can see here we've got a 4-bit structure with 4 ones, 2 ones, and no ones. And you can use this to write information. This is quite a small scale proof of concept, but in principle it works. Now, the issue is this fails consideration number 2. Assembled DNA hard drives shouldn't be overwritable at room temperature, because these ones definitely are. If you take your backbone, you add your staples, and then you write 4 separate structures, you add just one of the labels in each case, and you assemble them each separately, and then you mix them together, measure them. You should end up back with these, ideally, but what you instead get is mostly these here. So I did this experiment using this design, and here you can see the barcodes that I detected and the abundance with which I detected them. I mixed these 4 in equimolar quantities, so should be 25 in each of these. And you can see that it very much is not. It's primarily here and here. Most of them have all 1s or they're missing a 1. And we've heard a lot about some very nice error-correcting codes today, but I don't know if there is one that can fix this. All of your data ending up in the same spot is not ideal. So what are the solutions to this problem? How can we modify our scheme in the most simple possible way to not have them be overwritable? Well, you could, when you inject your strands, you could inject a 1, a biatylated strand in the case that you want a 1, or mutually exclusively, you can inject an identical strand but just without the biotin. So it won't be labeled by streptavidin and it won't produce a big enough nanopore signal to be visible. And in principle, this works. If you put a blocking strand in that's the same as this one but for the biotin, it isn't overwritten at room temperature. It's thermodynamically identical. It isn't replaced. But it does fail consideration number 1, unfortunately. N bits requires 2 N injectors, and what's worse, you're always only using N at a time. It's quite inefficient. So we can take a step back then and think about what the energy landscape of an ideal data storage module, an ideal one of these overhang systems should look like, and then see if we can realize it. In the first place, I'm going to start with an unwritten state, which is going to be a bare overhang, and a 0 state, which is separate, and which is lower in energy. So if you have the necessary material to make this, then it will form at equilibrium after the heating and cooling process. Now, we should also have a 1 state. It's only accessible if a writing oligo is present, so that gives you the chemical control over what you write. And it should also be lower in energy than our 0 state, so that at equilibrium, so after you've annealed and cooled down again, there is a free energy penalty for the 0 state relative to this one, and we mostly get the 1 state, which means that if the 1 state can be accessed, then it will be in the vast majority of cases. We also want, at room temperature, that the free energy landscape between 0 and 1 should be sufficiently difficult that it impedes transitions at room temperature. So you want your KBT to be kind of below the level of this energy landscape here so that overwriting doesn't occur. So these are our 2 considerations. The free energy gap between 0 and 1 should be big enough that there should be much more of 1 than 0 if the correct label is present. And we also want the energy landscape between 0 and 1 to be as obstructive as possible so that it can't be overwritten. And the scheme that worked, or that I found, was based on this idea of imperfect complements. So this is what an imperfect complement is what I'm showing it as here. It's identical to the actual complement to this overhang, but it's modified in some way. And by itself, the only thing that can form is this protected 0 state. I'll refer to these as protector strands hereafter. If you add your biatylated label at room temperature, even though it's a better complement, that kinetic energy problem means that it doesn't replace it, so it stays a 0. If you anneal this all together, then, as I said, the biatylated label wins because the 1 state is thermodynamically preferable. So this has the properties that we want. There's also the possibility that your protector strand is not perfect in one of those 2 ways that we talked about. That is, it's at room temperature, it actually is replaceable, so it can be displaced, which is something that can happen. And also, it can compete too effectively with the correct label oligo if that free energy difference isn't sufficiently large. And I tried various types of label, of protector strands to achieve this. But what our writing process should look like now is that we have in our basic oligo mix, we have everything we had before, but we also have these 4 protector oligos. Those are constitutively present, they're always there. And then we add our biatylated labels, and ideally what happens is we anneal it all together, and we still get our 0s and 1s in the right place. Because these labels are present, those protectors don't get a shot at binding to these overhangs. And I found through some experimentation, a few different ideas, that an effective protector strand is based on this idea of a single CT mismatch, a nucleotide mismatch. And a CT mismatch is particularly, an internal CT mismatch is particularly destabilizing for a DNA duplex. It creates this permanent loop. And the thermodynamic penalty is equivalent to about 3 base pairs, which is sufficient that if you do the annealing procedure, then this wins every time if it's available. And what's more, this permanent loop is not enough to allow a label to come in and displace this protector. They're pretty stable at room temperature, so I have here some data that I'll explain. So I've charted here the 1 to 0 and the 0 to 1 error. 1 to 0 error is the extent to which our protector outcompetes our label. So this is high if the free energy difference is not big enough. The 0 to 1 error is the rate at which a protector is displaced by one of these at room temperature in a space of 10 minutes here. And then there's the average error in a symbol, assuming that you have a distribution of equal ones and zeros. And the 1 mismatch case minimizes this error. It's about an order of magnitude lower than the control. Actually, this 10% 1 to 0 error here is not really indicative of how effective this scheme is. This is kind of a minimum baseline level, and this comes from just the labeling efficiency of streptavidin on our label. So in fact, if you use a different type of label which doesn't have this efficiency problem, this is probably pretty close to 0. It's at the baseline level. And even if you expose one of these to one of these protected overhangs to its correct label for 60 minutes at room temperature, you see effectively 0 to 1 error rate. There's effectively 0 displacement. So these work about as well as we could hope for. And I'll show you now an experiment I did using these protector strands on these 4 different overhangs. So I did the same thing as before, except this time I've included these protector strands, which are cartoonishly shown here with little locks. So you anneal everything together, and you have 4 different batches made in equal concentrations with a different label in each case. And then you mix them all together at room temperature, and you hope to get these, and this time you actually do get these. So this protector scheme is sufficient to protect these overhangs, but it also doesn't interfere with the correct writing procedure. So you expect to see something like 25% in each of these red boxes, and in fact you do, down to what is probably a pipetting error. And you don't see, notably and desirably, you don't see the big concentration of probability down here in the all ones pile. So, to conclude, we usually, in DNA origami and DNA nanotech, we try our hardest to avoid competition in our designs. We want to have one thermodynamic minimum, and we don't want anything else to be close to it, really, because that results in misfolding. But in this case, by carefully controlling the competition, we can actually help our structure to be more robust, especially, and it reduces the burden on our mixing procedure a little bit. So competition between DNA hybrids can be used to design more robust DNA data storage architecture. We've adapted our hard drive design here to the writing method, which helps reduce error, so we don't have to use as many error-correcting codes or as much logical or physical redundancy. But it also means that our writing device is easier to manufacture. And you can also use a very similar principle to this. Like I said, people in DNA nanotech are generally quite afraid of competition of strands, but you don't have to be, and in some cases it can help you. It can also be used to facilitate biosensing, for instance, if instead of looking for biotinylated labels, you're looking for 4 RNA markers or DNA markers, then in this case you could assemble a different structure in a whole population of different cells, and then you'd be able to freely mix together, measure that, and find out which markers coincided with which other ones, and you wouldn't have to worry about overwriting. So there are other applications of this kind of approach. So I'd like to make some acknowledgements to the lovely Kaiser Lab here at Ulrich's 50th birthday party, and also my master's student Justin Kronk, who helped me to take a lot of the measurements. And also I'd like to acknowledge funding source. My PhD was funded by DNA Fairylights, which is a Pathfinder project. Thank you for listening. And if there are any questions, I'll happily take them.
So thank you very much, Max. Do we have some questions, comments for Max? Okay.
Thank you for the talk. I've always wondered about DNA origami and in that how stable are the structures? So if I know obviously they're sensitive to temperature, but are they also sensitive, like the structures, if they're dried out, or do they dissociate in certain pHs or something? Or what other kinds of factors do you need to control to make sure the structures stay intact?
So you can dry them out pretty safely. I mean, you will dry them out. For instance, usually if you want to do AFM on them, for instance, you'll dry them out on mica, and it's not a problem. They stay intact, and you can resuspend them just fine. pH is an issue for DNA in general. You go too high or too low, and it will cause either degradation or it will cause it to break apart. But actually, interestingly, our nanopore measurements are done in a pH of 9.4, which is pretty high. It's above what you normally see for DNA nanostructures. You usually keep them around 8.1 or something with tris. But actually, there's a pretty decent operating window. They're not too delicate.
Any other question? Yeah.
Hey, thanks for the talk. The situation where you had the bit flips, so it sounds like what's happening is strands from one strand is displacing and then hopping onto others, something like that.
Yeah.
What is the energetic barrier? So these are basically, you're talking about kinetically trapped states, essentially, right? Is what you're trying to achieve. So what's different about the situation where you're using the protector? Why don't the protectors hop off and then your biatinylatids hop on?
Yeah, that's a good question. So in the case where you have a bare overhang, just a single-stranded overhang, the only free energy barrier is that you've gone from having 2 free species to 1 free species in a complex, basically. So there's an entropic hit there. In the case where you have a protector strand that's already bound, the intermediate in the kind of displacement there would be that you have both the protector, the incumbent strand, and also the invader, the label strand, both bound at the same time. But that usually requires some fraying of the protector that's already incumbent. So you'd require some base pairs, for instance, to detach. And there you're losing, for a second there, you're losing the number of base pairs that exist in the system, and that has a free energy cost. So removing something that's already bound will require some dissociation of base pairs and will always have a free energy penalty. I hope that answers the question.
Any other question for Max?
I have a question on scalability.
Okay.
Because you're using, if you decide to use origami, you're using a lot of bases for each bit, and so how would this kind of technology for DNA data storage be scalable, both in terms of costs, but also in terms of speed of writing and reading?
So in terms of cost, the only cost really here is the principal cost of making the oligos that you use to write information. The beauty of this is that you don't have to do any synthesis to make a specific data string here with a DNA origami structure. You have a pre-existing palette of oligos, and if, for instance, your structure is 100 bits, then you need, well, in this case, you'd need the 100 overhang oligos, you need 100 writing oligos and the 100 protector oligos, but you're never going to need to make any more than that. You just mix together the right ones to write whatever data you want. And you know, working with like a limited palette of DNA oligos that you can order in bulk, that is, has a cost benefit. In terms of writing speed, it's how fast you can mix things together. And so that's why microfluidics is a very interesting avenue here. You can generate droplets in droplet microfluidics at kilohertz rates, and if you can inject into them quickly and reliably enough, then that's kind of the upper bound on how quickly you can write these structures is a few kilohertz. In terms of reading speed, in nanopores like ours, these structures translocate completely in 1 to 2 milliseconds. So if your structure has 100 bits on it, then that's the upper bound there kind of for your reading speed.
I've got another question. You're using M13 as a scaffold.
Yep.
Do you consider that's the best choice, or maybe a bigger scaffold would allow you to encode more data? And is the way to go is to have many different M13 single strand where you put different staples, or would it be useful to have much bigger single strand sequence so that you could put even more data if it's compatible with your reading, of course?
I mean, a bigger scaffold would be actually quite beneficial, probably. So we could, for instance, use lambda DNA. It's better in the sense that you can fit more on it, certainly, but also because the speed of a free translocation in a nanopore is limited by, it's set by kind of the hydrodynamic forces. A longer molecule just has more drag on it, and therefore it takes longer per base to pass through the nanopore. It increases your temporal resolution because of that. So a bigger scaffold is actually quite useful, but probably something like lambda or M13, a viral genome, is beneficial for one thing because they tend to be pretty efficient. They don't tend to have too many repeated sequences, which do interfere with origami folding. And also you can, as we heard earlier, you can make a lot of phage. Making phage and producing phage genomes is pretty easy in scale. Yeah.
Thank you.
So any other question for Max? Okay, so otherwise, thank you very much, Max.
Thank you.